Hello everyone, I'm Phil Felgner, Director of the Vaccine R&D Center at UC Irvine in California. The title of my talk is Foundations of Vaccine and Lipid Nanoparticle Science. I'm using this timeline as an outline for my talk. Paul Ehrlich represents the period of infectious disease science that included Pasteur and Koch, when microorganisms responsible for infectious diseases were being discovered and understood. Attenuated organism technology developed then was adapted to the development of polio vaccine by Salk and Sabin, and 9 other vaccines developed by Maurice Hilleman. This was a real heyday for vaccine science. Alec Bangham is responsible for launching the liposome field. He mentored dozens of productive scientists who together were motivated by the promise of liposomal magic bullets, a term coined by Ehrlich 50 years earlier. For me, this talk begins in 1978 when I came to Tom Thompson's biophysics department in Charlottesville, Virginia, then to Syntex Research where lipofection was discovered, and Vical where Maurice Hilleman came to champion nucleic acid vaccination with us. I'll sum up with some recent COVID vaccine projects underway at UC Irvine. Here are a few things we learned in Charlottesville. We learned the exact dimensions and compositions of small unilamellar lipid vesicles. This physically stable self-assembled system is 20 nanometers in diameter and contains 2,500 lipid molecules with a combined molecular weight of 1.9 million kilodaltons. The exact dimensions of each lipid monomer in 3-dimensional space could be calculated with precision. This knowledge is useful to develop more advanced lipid nanoparticles today. The viscosity of the lipid bilayer can be a fluid like oil or a gel more like wax, depending on the chemistry and of lipid molecules, whether the aliphatic chains are saturated or unsaturated, and whether they are longer or shorter. The stability of lipid vesicles and nanoparticles is also controllable. Lipid vesicles composed of short-chain lipids disintegrate rapidly in contact with biological membranes, and nanoparticles comprised of long-chain lipids are stable for long periods. We also learned that the shape of lipid monomers determines the structure of self-assembled lipid nanoparticles. When the cross-sectional area of the polar head group is large compared to the aliphatic region, the cone-shaped monomers assemble into a micelle. When the space occupied by the polar head is equivalent to the aliphatic region, the cylindrical monomers form bilayers. Monomers that have a small polar head group and a large aliphatic region form inverted micelles that can organize into a hexagonal array. The mRNA vaccine lipid nanoparticles are lipid molecules contain lipid molecules with different shapes to produce a stable self-assembled structure. Some of this lipid nanoparticle biophysics was taken to Syntex Research in Palo Alto. Syntex was interested in immunology and to synthesize bacterial mimetic adjuvants for better vaccines. We produced a microfluidized squalene emulsion called Syntex Adjuvant Formulation 1. I also brought the idea to make positively charged lipid molecules with the correct shape to make stable bilayer lipid vesicles. There were many potential pharmaceutical uses for positively charged vesicles. One, to make artificial virus-like particles for gene delivery. Many of us studying liposomes envisioned making synthetic virus-like particles that could deliver functional DNA and RNA into cells for gene therapy. There was a technical problem standing in the way, illustrated here. The long dimension of the DNA was much longer than the internal diameter of the typical liposome. How can we get a 5,000 base pair negatively charged DNA strand that is 1,700 nanometers long into a 70 nanometer bilayer lipid vesicle? What if we mix the cationic liposome with negatively charged DNA and capture all of the DNA into a lipid aggregate? We expected positively charged liposomes mixed with negatively charged DNA would form a visible aggregate, and we got our first surprise. They didn't produce a visible aggregate. To prove that they interact, we used sucrose density gradient centrifugation. The lipid, cationic lipid, made all of the DNA float, proving that they interact. Surprise number 2, it worked right away in transfection assays. Using a very simple protocol, these lipoplexes deliver functional DNA and mRNA into cultured cells, producing the encoded protein product. Shown here is the first experiment on 2 different cell types. Cells produced chloramphenicol acetyltransferase enzyme after transfection of the gene. We developed a high-throughput transfection assay, screened hundreds of lipid molecules and thousands of optimized formulations to arrive at the 1987 lipofection paper in PNAS. Rick Roman, the chemist who first made the prototypical cationic lipid, DATMA, saved his lab book entry dated 7/5/84. He retired recently from Thermo Fisher and is now a cobbler making designer shoes for $5,000 a pair. What are these cationic lipid complexes we call lipoplex? The top is a negatively stained EM image showing a field of regular shaped 80 nanometer particles in a background of filaments. None of these structures are evident when either the cationic liposomes or DNA are imaged alone. Below are atomic force microscopy images showing plasmid DNA on the left, a field of 80 nanometer lipoplexes in the middle, and one lipoplex zoomed in to the right. This particular lipoplex is a condensate of a single individual 5,000 base pair plasmid. We used space-filling molecular modeling to show DATMA dope lipids, how they organize in the presence of DNA to form the lipoplex. The modeling used the lipid packing parameters that were established by Jeff Mason and others 15 years earlier for small unilamellar liposomes and other lipid nanoparticles. It shows a 10 nanometer DNA strand in the center of an inverted micelle. According to this model, one 5,000 base pair plasmid DNA can be condensed into one 58 nanometer lipid nanoparticle surrounded by a lipid bilayer. This structure resembles mRNA vaccine lipid nanoparticles that can be imaged by cryo-electron microscopy on the next slide. On the right are 70 nanometer mRNA lipid nanoparticles. On the left is a negative stained EM of small unilamellar lipid vesicles. The vesicles on the left are empty, and the ones on the right are packed with mRNA. They can be frozen and thawed and are remarkably stable. The assembly is so good as though the RNA is a scaffold holding it together. We took some of these results to Syntex for their support, and they declined, saying gene therapy is for the year 2020. So we joined with UC San Diego scientists to start up a company called Vical. There, in collaboration with John Wolfe from the University of Wisconsin, we got our third surprise, that DNA and messenger RNA injected intramuscularly is functional, expressing reporter genes at a level comparable to what we could get in cultured cells. The mRNA for this study was made by following methods taken from Doug Milton's two beautiful papers published in 1984, the same year that DATMA was first synthesized. Although there are many potential applications of this finding, we prioritized vaccination. With Maurice Hilleman supporting us through Merck, we demonstrated heterologous protection against influenza by immunizing with DNA encoding the nucleocapsid protein. Much clinical development in nucleic acid immunization was accomplished at ViCal, but success never reached the pinnacle that it did today, largely because of the challenging cancer and viral targets that we prioritized then, and because we didn't have the range of beautiful lipid nanoparticles that we have today. Then at UCI, we developed an approach for the discovery of vaccine antigens. We developed a protein microarray platform that can be used to make all of the individual proteins from any microorganism. The arrays are used to identify antibodies that are induced after infection. Since 2005, we made all of the proteins from 38 infectious microorganisms, printed 100,000 arrays, and probed 50,000 serum samples collected worldwide. Then in March 2020, we were prepared with a coronavirus antigen microarray that is used to measure antibodies in people who are infected with SARS-CoV-2. Here is the array containing proteins from SARS-CoV-2, SARS, MERS, common coronavirus, and influenza. The images show how the array looks when it has been exposed to serum from people who were either negative, infected with SARS-CoV-2, or vaccinated with the mRNA vaccine. The intensity of the spots increases from left to right, indicating the increase in antibody levels in the samples. Our lab was supported during the outbreak to conduct COVID research and surveillance when most of the campus was shut down. We probed 10,000 fingerstick blood specimens collected from residents throughout Orange County and from the UCI Medical Center. In December 2020, herd immunity was climbing due to the natural exposure to virus in the environment. Then, on December 16, 2020, 6,000 doses of Moderna mRNA vaccine were administered to healthcare workers at the UCI Medical Center. And the seropositivity rate in that population shot up to 78% a month later. We didn't believe the results, but in February it reached 93%, and by March it topped 99%, and we were believers. The mRNA vaccine induces spectacular immune responses. Where do we go from here? Our program in vaccine science has two research categories. Under vaccine discovery and development, we prioritize from a list of viruses, bacteria, parasites, and cancer to select vaccine antigens and test DNA, mRNA, and protein vaccines for efficacy in animal models. We need experts in all of the topics we engage. We have animal high containment facilities at UCI and have approval to conduct select agent research. In parallel, we study immunological mechanisms and lipid structure-function relationships with the goal to make more effective vaccines with less reactogenicity. With that, I'd like to thank the members of my team and the laboratories of Hugh Davies and Lisa Wigar in the Vaccine R&D Center. I also want to thank our funders at DITRA, the NIH, and our corporate sponsor, Polaris Pharmaceuticals. Thank you very much.